My name is Will Ludington. I'm a scientist in the Biosphere Sciences and Engineering Department at the Carnegie Institution for Science. My lab works on the gut microbiome, and we use the model system of fruit flies to try to understand how the specific bacteria colonize the gut and how they interact with each other to influence the ecological properties of the microbiome. Our gut is full of bacteria. It's got the diversity of the Amazon rainforest. We're using a simplified system in fruit flies where they have a gut diversity of about 5 species. So we can really understand at the molecular level what the evolutionary adaptations are of individual species. We can turn individual genes on and off when we want in individual cell types throughout the fruit fly's body. My team is just really interested in getting into the biological details, just really trying to get an intuition for the system, really try to understand how each little piece works and how they work together to create the whole. And so we're really interested in developing techniques, new techniques that we can use to gain new insights into the biology of these organisms that we study. Raising the flies is a really big job. It's easy to do, but it takes a lot of effort and a lot of really precise care. Rajan Just is the manager of our fly facility, and she does all the very careful preparations of the materials. Ren Dodge is our in-house videographer. He's developed this technique where he can see the individual bacteria in the fruit fly's gut. This is this amazing amount of biological detail that we can capture. So we can look inside of an individual fly and see these bacteria as they arrive inside. And watch what they do. Angela Hsu is a graduate student in the lab, and she's trying to understand how the bacteria are influencing the aging of the fly and the lifespan. If they take the microbiome from an older animal that's got neurodegeneration and they put it into a young animal, the young animal also would get that neurodegeneration, and they'll seem old, they'll be frail. The reverse also works, and that's the really cool thing. If you take the microbiome of a young, healthy animal and put it into an animal that has got neurodegeneration, but first you clear out the old animal's microbiome, the fresh, young microbiome is like fountain of youth. Kevin Miller is another graduate student, and he's working on the molecules that the bacteria use to stick to the fly's gut. We've found the key to the molecular Velcro that these bacteria are using to find the specific spot inside the gut. And what's really interesting is that it's very specific. So it's not like they can stick to anywhere in the gut. And modifications to those molecules we expect would actually change where they would go inside the animal. Karina Gutierrez Garcia is a postdoc in the lab, and she uses computational approaches to study the evolution of the bacteria and the way they are binding and finding the niche inside the fly. Robert Scheffler is a postdoc in the lab, and he's studying how bacteria sense their environment and really specifically sense other strains in the environment. So it's really interesting that these bacteria, if you give them the perfect growth conditions, they won't really grow until they know that their partner species is present. Julie Breyer is a master's student, newly arrived, and she is working with Robert right now in order to really characterize the genes that are involved in this sensing system. Josh Derrick is another graduate student in the lab, and he's interested in how we uptake lipids from our diet. The really interesting observation is that if an animal has bacteria in its gut, it has an easier time of taking up bacteria than if it's germ-free. So Josh has found the same thing in fruit flies, and he's using the incredible genetic tools we have in the fly in order to investigate these uptake mechanisms and how they are changed in response to the bacteria. Haolong Zhu is a graduate student and is working on really figuring out the specific biology of the niche where the bacteria go inside of the fruit fly's gut. Haolong has figured out the specific genes that are most critical for individual cell types within this niche tissue. And now we can really investigate specifically in this niche tissue, what are the cell types and how are individual genes responsible for the colonization phenotype inside the fly. All of this is happening in this lab on a daily basis.